Hi everyone, good morning. And yes, since it's a final day, I would also like to take the opportunity on behalf of my co-chair Liz Harvey, who isn't with us, but on behalf of the award committee, to thank everyone that had a role in this process, from the nominators, the writers of the support letters, the subcommittees and the chairs, and the ASLO communications and business offices, for all the hard work. It is a hard process, and I urge you to be involved in it and either nominate a friend, a colleague, be involved in the subcommittees. There are lots of ways to be involved, so we're waiting for volunteers. And personally, I would really also like to thank the awardees that throughout this week have shared with us their inspiring stories and experiences. For me, it was a real honor to get to know you and some of what you have done over the years. And with this, I would like to turn to this year's award. And this is a John H. Martin Award, which recognizes a paper in aquatic sciences between 10 and 30 years old. The paper is 10 and 30 years old, hopefully not the awardee. And this paper is judged to have a high impact on subsequent research in the field. And this year, and in this beautiful spot, limnology and lakes are given special center stage via the award presented today. And the 2024 Martin Award is presented to Hanson, Bade, Carpenter, and Kratz from 2003. It's an L&O paper, so a society paper, lake metabolism, relationships with dissolved organic carbon and phosphorus, for showing that lakes experience seasonal net heterotrophy, except in cases of high phosphorus and high primary production. In this paper, Hanson et al. Used in situ sensors to measure dissolved CO2 and oxygen and combined that data with dynamic system models to produce continuous estimates of surface water metabolism over extended periods in 25 lakes. The work fundamentally advanced the understanding of ecosystem metabolism in lakes and the balance between production and autotrophy and heterotrophy or respiration, finding that lakes experience seasonal net heterotrophy. This publication has influenced the field in the years since its publication, with avenues of research opening in lake metabolism, carbon budgets, and long-term observational networks, including the Global Lake Ecological Observatory Network, LEON, that began in 2005, if I'm not mistaken, and is really stimulating new approaches in limnology and new tools all over the world, with scientists all over the world participating in GLEON. I'm quoting from the nomination letter. This work underpins a remarkable transformation in the field of limnology, based on the technologies of lake sensors and the quantitative models used to interpret data. And it is especially appropriate to present the award here in Madison, Wisconsin, to a local scientist from the Center of Limnology. I am pleased to introduce Dr. Paul Hanson to receive the award on behalf of the co-authors.
And again, it is my sincere pleasure to award the John Martin Award to Paul Hanson on behalf of myself, the ASLO board, and the hard-working awards committee and subcommittee. So with sincere congratulations.
Well, thank you for that wonderful introduction. Wow. And thank you all for showing up for the Friday plenary this week. It's been an absolute amazing week. And gosh, what an honor for Darren and Steve and Tim and me to have our paper selected for the Martin Award. And as Alana pointed out during this week when we have ASLO here in Madison. And it's been just a fantastic week, seeing a bunch of old friends and meeting and making a lot of new friends. So it's been terrific science and terrific camaraderie and just a wonderful week. This paper on lake metabolism was the first chapter in my dissertation. And it was a lot of work. And I was so proud to have it published in L&O when it came out. And I remember going back to Steve Carpenter, who was my advisor at the time, and so Darren and I were both in graduate school, and Steve was our advisor. And remarking to Steve that I couldn't believe how much work it is to actually write a paper and try to get it published. And his response was, "Yep, don't worry about it. After about 20 papers, you'll start to get the hang of it." So, so still going. And what an honor to be listed amongst the recipients of the Martin Award. I'd never had the pleasure of meeting John Martin, but his legacy is here with us today for sure. And the amazing work he'd done in oceanography, and specifically the primary production in oceans stimulated by iron is related somewhat to what we'll be talking about today. So on behalf of all the co-authors, thank you to ASLO, and thank you to the ASLO committee for selecting our paper and giving us the opportunity to talk about it and talk about lake metabolism. I also thought it'd be fun to do a quick, where are they today, of the co-authors. Darren Beatty, other than sitting right here, is at Kent State as a faculty member doing his thing there. Tim Kratz, who was the director of the Trout Lake Station when this work was going on in 2000, is retired and allegedly in the Madison area. I haven't seen him locally for a while, though. And Steve Carpenter has become one with ecosystems, I think, as a free-range scientist. And I did hear him mention the other day, we have to bring people and ecosystems together. So I think he's achieved that. And that's me in the lower right. And I put it there as a reminder that sometimes the nut doesn't fall too far from the tree. And I hope you'll see, and I think you will see, some of Steve's influence throughout the talk. Oh boy, a lot of acknowledgments to make for this. ASLO for the Martin Award and for the generous support for attending the meeting. A whole host of people from the Center for Limnology, including my contemporary metabolism co-conspirators, Hillary, Robert, Noah Lottig, Ellen Albright, Bennett McAfee, Sophia Skoglund. The North Temperate Lakes LTER run currently by Emily Stanley, and all of their staff, including Noah Lottig, Mark Gaylor, and Paul Schramm, who run the buoy systems, were incredibly important to the data and the science that went into this. We'll talk a little bit more about LTR in a little bit. And of course, colleagues and friends at the Center for Limnology, including Jake and Gretchen and Grace and Olaf and John, and graduate students and postdocs, past and present, who have really pushed this forward. And I'd also like to thank friends and colleagues from the Global Lake Ecological Observatory Network, and most recently, especially the Gleon Fellows, who we'll talk a little bit about too. So funding was provided by ASLO, the U.S. National Science Foundation, and historically UW-Madison and the Gordon and Betty Moore Foundation as well. There's no way in a 30-minute talk I can do justice to all the amazing work that's been done in metabolism out there, and what it's done to actually lead the field forward and to influence me and to influence a lot of the stuff you're going to hear about today. But I did want to bring up a few programs and folks. The Grill colleagues in Montreal, especially Paul, Eve, and Dominic Vachon. Trophic Cascade, the late great John Cole and Mike Pace, Steve Sadro and his Mountain of Metabolism literature, Kaylin Carey's group at Virginia Tech, and all the graduate students and postdocs who over the years have been working on metabolism and currently are. The Lake Sunapee group, including Kathy Weathers, Dave Richardson, Denise Brezowitz, and the Wizards of Oz, David Hamilton, Justin Brooks, Matt Hipsey, and USGS crew. Especially Jordan Reed, Emily Reed, Jake Swart, Sam Oliver, Allison Appling. And then, if you haven't read it, you got to check out Kathy Jo Jankowski's amazing literature review on metabolism where I also go to harvest ideas about metabolism after these many years, and many, many more. Okay, into metabolism. So lakes have metabolism. That's a question I had when I started as a graduate student. From a systems perspective, it's really helpful to think of the conceptual model of lake metabolism in managing organic carbon and other aspects of water and water quality in lakes. It's a way in which lakes can receive organic carbon and process it, create organic carbon within lakes, bury it in the sediments, mineralize it, send it back to the atmosphere as carbon dioxide, or pass it along to downstream ecosystems. And we all know how important organic carbon is for lakes, everything from food webs to water quality. But the processing is also really important because it helps determine the role that lakes play in global carbon cycles. For example, the emission of greenhouse gases from lakes and the massive storage of organic carbon at the global scale that's in the sediment of lakes. I'm not going to be talking about global carbon budgets today, but rather focusing very much on lakes as ecosystems and trying to understand how viewing those ecosystems through the lens of lake metabolism helps us address a number of interesting and important questions. Let's start first from a perspective of organic carbon. This will be familiar to all of you. Organic carbon is available to lakes both from external sources, which we call allochthonous organic carbon, and internal sources. And I've put up here pictures of two sort of iconic types of lakes. Dystrophic lake, which in this case is Crystal Bog from northern Wisconsin, and a eutrophic lake, Lake Pettenwell from central Wisconsin. And I put them up here so we could sort of compare and contrast a little bit the importance of these different sources of organic matter. It's well established that bog lakes and small dystrophic lakes receive a lot of external carbon from surrounding peat and other aspects of the terrestrial system is responsible for their kind of brown color. Eutrophic lakes also receive external organic carbon, but relative to internal sources, it tends to be kind of small. The internal sources, of course, are very high in eutrophic systems, and we call this the autochthony for those systems. And that includes all the plant biomass and microbial biomass in a certain sense, too. And for eutrophic systems, of course, it's the external nutrient inputs that are driving this from surrounding catchments, which is really high in eutrophic systems, and can be higher or moderate or even low in dystrophic systems, and of course in oligotrophic systems too. And this plays out in the color of these different lakes. So brown, of course, for dystrophic lakes, and more green for eutrophic lakes. Now keep this in mind as we move on to some of the details of metabolism. This is going to kind of set us up for the paper that we won the award for and kind of be the launch point for how we move forward. So this is a simplified model of a lake and lake metabolism represented here by a classic box and arrow diagram with the boxes being state variables that we can go out and measure typically, like inorganic carbon or carbon dioxide, dissolved oxygen, organic carbon. And the arrows represent the processes. How do we make those transformations between the states in the system? Those processes are primary production, so that's responsible for the autochthony that's happening in lakes, and respiration, which takes that organic carbon that's produced and mineralizes it and turns it back into carbon dioxide. In the process of primary production, of course, we produce extra oxygen, and respiration consumes that oxygen. So we have this continual cycle that's going on in lakes. This was described well a long time ago by H.T. Odom with respect to changes in oxygen. So the change in oxygen is a function of the primary production minus the respiration, plus the atmospheric exchange, and a lot of other things that we try to ignore, but as you'll see, will come into play. So what does this have to do with the organic carbon we were just talking about? It's important here to introduce another concept, which is net ecosystem production, the difference between primary production and respiration. And if primary production is high and it's greater than respiration, then net ecosystem production is high. It also means we're producing organic carbon, we're producing excess oxygen, and we're consuming inorganic carbon, which is typically CO2. And conversely, if respiration is greater than primary production, net ecosystem production is negative, we're consuming organic carbon, we're consuming dissolved oxygen, and we're producing CO2. Now, these are the basics of metabolism, and they underlie the development of the technology and the evolution of the ideas that have gone into using metabolism for a lot of different purposes. But the question remains so far, how do we actually measure this and quantify it? We're really interested in quantifying those rates, because if we can quantify those rates, we now can unlock the power of the model to apply this kind of approach to all kinds of different questions. We can try to understand how external forcing in the system might change productivity and the production of organic matter. We can study how changes in external loads and nutrients can affect the system. Measuring metabolism, at least back in 2000, really meant measuring dissolved oxygen and dissolved gases in the system. And if you measure those at high enough frequency for a long enough period of time, you can see the overnight changes, where during the day when sunlight is affecting the system, we have primary production and increase in dissolved oxygen, and at night we have the decrease in dissolved oxygen due to respiration. But over longer time periods, we can also have persistent and elevated primary production due to external phosphorus loads and persistent elevated respiration due to external loads of organic carbon. Okay, so onto the paper. We wanted to know how phosphorus and DOC controlled metabolism balance in north temperate lakes. And so we looked across gradients that are relevant to what we've just been talking about. We couldn't in this study at least measure the external inputs of phosphorus and organic matter, so we assumed that they were related to the concentrations you could find in lakes in northern Wisconsin. And so we found 25 lakes that cross broad gradients in dissolved organic carbon and total phosphorus. We put together a buoy, a collection of small buoys that had CO2 equilibration chambers and ergas to measure the dissolved gas. And we hung YSI DO sondes off the side of the buoys. And we stuck them in the lake for a few days and continuously measured the dissolved gases and did the biogeochemical workup at the same time. And this is the only result I'm going to show from that paper. On the left, you've got the time series of dissolved oxygen and carbon dioxide for 3 different lakes over a few different days. And if you look at the bottom figure of Crystal Lake, I think it shows it most clearly that during the daytime, you do indeed have that increase in dissolved oxygen and decrease in CO2 that's due to primary production in the system. And at night, it's just the opposite. We're consuming oxygen and producing carbon dioxide. Those are turned into the estimates of respiration and primary production that you see on the right. And the top 2 panels show respiration as a function of DOC on the left. And you see that as DOC goes up, respiration goes up as well. And on the right, as total phosphorus goes up, primary production goes up. So that's all good. And net ecosystem production makes this a little bit more interesting. So what's the balance of the system? Are we producing excess organic carbon and oxygen, or are we consuming? And across the total phosphorus in the bottom right, it's not quite so clear. At low total phosphorus concentrations, it's a little bit all over the place, but tends to be near imbalance or slightly negative. But at very high phosphorus concentrations, it's positive. And DOC in the lower left, it's a little bit more clear. As DOC concentrations increase, we tend to go negative in net ecosystem production. So we learned a number of things from this paper. Low nutrient lakes are nearly in metabolic balance. High nutrient lakes, and it's got to be pretty high P, have primary production in excess of respiration. For DOC, it's a little bit different. Once you get above about 10 milligrams per liter, you tend to have high respiration rates in those systems. And measuring dissolved oxygen is much easier than measuring carbon dioxide. And that's still true today, although that's starting to change too. But just as importantly, we showed that the technology works for making those dissolved gas measurements. The models work that we put together to turn those measurements into metabolism estimates. And we felt like the ideas really had legs. So we kind of felt like we're off to the races. We have a really good start into this world of lake metabolism. But this study was just the surface water of lakes over a few days, and we all know lakes are a lot more complicated. And if you were to look at things over a longer time period or at a more whole ecosystem perspective, you'd probably get different answers. And so let's look at this. You're a bunch of trained limnologists, and you will automatically see a bunch of things with your eyes that have to do with the physics and the biology of the system. These are data off of Lake Mendota in 2009 during the ice open period. And the top panel shows dissolved oxygen. It shows a measured dissolved oxygen in black and what it would be if it were at equilibrium with the atmosphere in red. And the bottom is temperature. And temperature is shown primarily to help you understand when mixus occurs in the spring and stratification and then mixus again in the fall. And it's pretty clear that things change throughout the season. And I think it's helpful to look at the dissolved oxygen really as a function of temperature, because temperature is a master variable in the system. And when we plot those against each other, we see dissolved oxygen going through this annual cycle that has a lot of hysteresis in it. It starts in the upper left in the spring, being relatively high compared to what it should be at saturation based on temperature. And that's when the ice breaks up. Diatoms and green algae are going crazy, and there's a lot of primary production in the surface water producing oxygen in the system. Shortly after stratification in the spring, however, everything calms down, and that in Lake Mendota is the clear water phase, and you don't see a lot of dynamics in the dissolved oxygen. But it doesn't take long, and Trina McMahon will tell you this, that when the water turns about 18 degrees Celsius, the microbial community wakes up, and specifically blue-green algae, and this year was no exception. We've had big Phanosominum blooms that started at about 18 degrees Celsius. That carries on for a little bit until we hit 20 degrees Celsius, and then everything goes crazy as the cyanos take over the system, and we are in the algal-dominated phase. And that goes on for the bulk of the summer and into the autumn. Then all of a sudden things change and dissolved oxygen goes negative. And so it seems like we go from this period of positive net ecosystem production to negative net ecosystem production overnight. But obviously there's physics at play. And once we get into fall mixes, the lake is struggling to get back to the equilibrium concentrations. So there's a lot embedded in here, and as ecologists we know that it's not just primary production and respiration, but there's a lot of physical processes that are going on. And so I think it's helpful to think about the evolution of the science and the technology together, and I put it up here as kind of three major steps that we've gone through in the metabolism world. The first was just trying to understand that balance and develop the technology to quantify the balance between primary production and respiration. And there's about a decade of pretty intensive work that went into that. The second is the ledger. So using metabolism as a process to get at the carbon balance in the system. How much is coming in? How's it being processed? What are the fates and where is it ending up? And most recently, thinking of metabolism as an ecosystem engine of water quality. How is this driving the system forward? And what are the changes that we can understand in water quality based on this? But note also that it's all of this is happening within a backdrop of the scientists and the people who are collecting the data and moving the science forward, as well as the development of the technology. And I want to use that to move into GLEON here for just a few minutes. So GLEON is a Global Lake Ecological Observatory Network. Founded in 2005 by a couple of limnologists, Tim Kratz and David Hamilton, and a couple of computer scientists, Peter Arsberger and Fang Peng Lin. And they posed this question to the community. Would it be helpful to develop a network of observatories to address this question in the upper right of new scales of observation, creating new opportunities for new science questions and new modeling approaches? And the lake community thought, yeah, it seems like a great idea. And so the approach was, okay, well, maybe we can replicate these observatories all over the world. We'll have new gradients and lots of new data, and we'll be off to the races. Well, what happened, of course, was there was a lot of differences in how each one of the countries and the members developed their instrumentation because of local funding and local needs that really provided the resources to create that. But this turned out to actually foreshadow GLEON's greatest strength, which is its diversity. Its diversity not just in equipment, but of course the human diversity and the human capacity that's brought to the table. But there were a lot of papers produced in GLEON in that first 10 years around metabolism. I've just put up a few here that kind of represent a little bit of the global diversity. Peter Steyer wrote a great paper on the methodology and what the state of the science was Peter from Denmark. Metabolism in small lakes in Taiwan was super cool as typhoons come in and essentially replace the entire volume of the lake in about 1 day. Seasonal switching between autotrophy and heterotrophy in Estonia by ASLO and his group showed that it matters a lot when you measure metabolism in lakes. Heterogeneity in Brazil, a lot of papers on spatial heterogeneity in the US as well, and in northern Wisconsin. And how remediation of lakes actually has an effect on metabolism. So those are just a few examples, and there are many, many more papers too. But I do want to call out this particular paper by Chris Solomon, where he brought together data from 50 different lakes and did the metabolism estimates. And there are two important things to show here. One is that the slope of the relationship between respiration and primary production is pretty solid, which suggests that the primary production produced in the lake is probably very labile and is consumed easily by microbes that are in the lake. But if you get rid of that primary production, you're still left with some remnant respiration, and that is related to the external loads of organic carbon in the system. Well, GLIAN evolved quite a lot, and it has become really more of a framework for collaboration, taking new steps into team and team science, and that's reflected also in a fellowship program that Kathy Weathers and I put together about 10 years ago to focus on developing teams who can attack these big scale, difficult problems that have to do with big data volumes and modeling, and using team science approaches to get there. And some of these teams actually adopted metabolism as part of the work that they were doing. And here are 3 examples. In the first cohort, Hilary Dugan and others took a look at the kind of modeling that was done in metabolism and the flux models for atmospheric exchange, and determined that it really has a big impact on how you interpret what happens. Ian McCullough and colleagues modeled 5 different lakes, and I'm just showing Lake Monona here, for over 5 years. So now we're getting into longer term data rather than shorter term data from these buoys. And also discovered that the fate of the organic carbon in the lake depends a lot on the time of year and the nature of the lake, with some lakes respiring almost all their organic carbon in the summer and bearing a lot in the winter, depending upon trophic state. And the current cohort number 5, is linking satellite remote sensing to ecosystem function through the microbial community and ecosystem metabolism. So that's pretty cool. But all of those projects required new ideas and new steps into lake metabolism that really have kind of expanded the ideas and required a little bit more comprehensive ecosystem approach to understanding what's going on. And to take the next steps, I want to switch back here to Wisconsin and look a little bit more at the North Temperate Lakes LTER. So it's been important here as well. And give a big shout out to Emily Stanley, the current lead PI of LTER, as well as all the staff who have made the data gathering and availability possible. It's really a remarkable program that has put together more than 40 years worth of data and science. And we've leaned heavily into these data to be able to understand how ecosystems are changing through time. That's true for metabolism too. And I'm just going to show 2 quick examples. The first is by Austin Delaney, who wanted to know the influence of carbon cycling on oxygen depletion, specifically on hypolimnetic oxygen depletion. And so he modeled up, using those data, those 40 years of data, 6 of the LTER lakes. And what he discovered was that autochthony in general greatly exceeds allochthony, but almost all of that autochthony gets consumed before it can contribute to storage in the system. And that means that it's also hard to measure because when it gets produced, it also gets consumed shortly thereafter. Eutrophic lakes are a little bit different because they produce so much excess organic carbon. A lot of that gets exported from the surface waters into lower depths and gets consumed, creating hypolimnetic anoxia. And typically that's done in such excess that we build up this reserve of organic carbon in the sediments, which is legacy organic carbon that can go on and fuel hypolimnetic oxygen depletion well into the future. Well, this gave us the bright idea that, we could take this water quality bit a little bit further and look at other aspects of water quality besides hypolimnetic depletion. And so we recently published a paper on legacy phosphorus and ecosystem memory control. It's really metabolism linking phosphorus cycling and organic carbon cycling in Lake Mendota over a long period of time. Oop, that one had a timer on it. And so we had to change our thinking, expand it a little bit in lake metabolism to link two now pretty well-known cycles, the phosphorus cycle in lakes and the organic carbon cycle in lakes. But we also had to add a sediment component to it too, because we recognize the importance of the buildup of phosphorus in the sediments over the 150 years of development in the Yahara watershed that affects Lake Mendota. And so what did we do with this model? We took advantage of the LTR data and we calibrated it to 40 years of LTR data and reproduce the historical water quality. And then we're able to go into scenario mode and simulation mode and turn off that external phosphorus input and see what happens. And this is what happens. So if you look on the right, that is the recovery of a number of different variables. And I have three variables of interest here: epilimnetic phosphorus, water clarity is measured by Secchi depth, and then hypolimnetic anoxia, the number of days of hypolimnetic anoxia. When you turn off the phosphorus, I don't know if you can see the little blue arrow. Oh, good. Okay. You get an immediate flushing that's based on the hydrologic residence time of the system, and so phosphorus does go down in the epilimnion. But then there's a long, slow decay in phosphorus that really is due to the release or the internal loading of phosphorus from that built-up legacy phosphorus that's in the sediments. And that just takes a long time, about 50 years. To kind of either bury or eliminate from the system so you're back to a more pristine state. For water clarity, you do get an initial bump, but summertime Secchi depth still remains relatively shallow for a long time until you hit an inflection point a little after 50 years, and it takes until about 85 years into the future before you see more pristine conditions for summertime Secchi depth. And similarly for hypolimnetic anoxia. You hit that inflection point about 50 years in, and it's not until about 100 years in that you actually reach recovery of the system. And so what we found is that recovery times of these important water quality variables vary depending upon which variable you're looking at, and that there's a dependency between these two in a sequence: phosphorus, then clarity, then anoxia. So there's a big legacy of the 150 years of phosphorus that was put into the system and ecosystem memory because of the linkages through metabolism in the system. So, quick summary of what we've been through. Under what circumstances are lakes that are heterotrophic versus autotrophic? Well, the trends tend to be scale specific, but from a carbon budget perspective, of course, when allochthony is greater than autochthony, it is net heterotrophic. But it's more fun to actually think about the organic carbon itself and how it's processed and what it means for the system. What roles do they play, lakes play in the carbon cycle? Well, they store a lot of carbon, but they also mineralize carbon in various forms and send it back to the atmosphere as CO2 and methane in a lot of cases. And how do the nutrient legacies of ecosystem memory control water quality? Well, conservative nutrients like phosphorus stay in the system a long time, and it makes it really clear it's much easier to break an ecosystem than it is to fix it. And we've learned through long-term ecological records, like from the NTL LTER, that this can take a long time. I just have three super quick slides on looking at the future, and if I were a young scientist interested in pursuing these things, this is probably where I'd go. Legacies and ecosystem memory, we looked at this on one lake, under one set of conditions. There's a lot of work to be done here on how our system's going to change into the future and how quickly can that happen. Scaling, can we take what we've learned from the few well-studied systems that we have around the world and extrapolate that to places around the globe that are desperate to have better understanding of their water quality futures? And of course, we're in Wisconsin. It wouldn't be a Wisconsin talk without a cow. And what effect do extreme events and fat-tailed distributions have on what we understand in the system? Metabolism models tend to be pretty smooth, so maybe it's a matter of what can we not explain with a metabolism model and pick up that unexplained variance as new information and new ideas to drive us forward. So thank you very much for listening to our talk today. And once again, I want to thank the co-authors for this great paper and what it's really meant for all of us. Thank you. I think we're out of time, so I'm going to be around all afternoon, and I'd love to meet and address any questions that you might have. Thank you.